My name is Simon Warren. On behalf of my colleagues at Indiana University, I'd like to present a study on Xanthogranuloma, which shows that NTRK fusions are a common driver in the solitary variant. Here are my disclosures. This figure shows the range of tumours that have an NTRK fusion as the initiating driver, classified by frequency. In red are tumours where more than 90% of the cases are associated with an NTRK driver. Turning to the NTRK molecule, this figure shows its structure and function and applies to NTRK1 through NTRK3. It's a transmembrane receptor for neurotrophins. Here is the cell membrane, and here is a neurotrophin binding to NTRK. Neurotrophins are homodimers. When they bind to NTRK, the NTRK molecule itself homodimerizes, and this results in transactivation of the kinase domain, and then downstream activation of the MAP kinase pathway, the protein kinase C pathway, and also the PI3K mTOR pathway. Turning to Xanthogranuloma, it's the most common category of histiocytic neoplasia. Most cases are solitary. Most solitary lesions resolve spontaneously, but there's a range of clinical behaviours with multiple cutaneous lesions and less frequently evolution to disseminated disease. Eye and brain involvement confer significant morbidity, and mortality can be 25% in disseminated disease. We encountered 2 cases of solitary Xanthogranuloma with NTRK fusion. In both, we showed strong reactivity with pan-NTRK by immunohistochemistry. Because these were 2 sequential patients, we wondered if NTRK fusions were much more common in solitary Xanthogranulomas than previously recognized. So we worked on establishing an NTRK immunostain for histiocytic neoplasia. We used an NTRK fusion soft tissue tumor with a known NTRK fusion as our positive control. We obtained negative results in 14 cases of reactive histiocytic processes. This is sarcoid. We obtained negative results in 5 cases of Xanthogranuloma with known non-NTRK drivers. Panel C is a disseminated Xanthogranuloma with BRAF fusion driver, and panel D is a disseminated Xanthogranuloma with PTP N11 point mutation driver. We then screened a total of 55 Xanthogranulomas using this NTRK immunostain. We scored them as follows: 3+, 2+, 1+, and negative. Here are our results. We identified 26 cases with positive immunoreactivity out of 55, but these were all restricted to solitary Xanthogranulomas and none in the disseminated Xanthogranulomas. We sequenced a subset of 23 Xanthogranulomas. In the top row are the immunostain results, with positive in red and negative in green. In the next row are the DNA and RNA sequencing results. So all the cases that were positive in the immunostain, even the 1+ scores, we were able to detect a fusion in blue. Fusion partners are written in black. All the cases that were negative in the immunostain lacked a fusion, and that's in grey. The lower portion of this table shows other recurrent mutations that we identified. Recurrent meaning 2 or more cases. I don't have time to go into these, but the theme is that they're synergistic with NTRK and contribute to downstream activation in the mTOR, PI3K, MAP kinase pathways that are downstream from NTRK, suggesting that an NTRK fusion alone is not sufficient to produce even a solitary Xanthogranuloma. We also looked at NTRK RNA expression. In this figure, the cases are subdivided with NTRK fusion in blue and wild type in brown. There is a mean 58-fold difference in expression in the fusion cases, meaning that the promoter from the fusion partner contributes to overexpression of the fusion RNA in all cases. This table shows which exons from the fusion partner and NTRK are present in the fusion protein. So for example, in cases 1, 5, 9 and 10, there are exons 1 to 8 of the fusion partner TPM3 and exons 12 to 17 of NTRK, which provide the NTRK kinase domain. This shows that in all our cases, there's a second method of activation of NTRK, since the fusion partner provides a zinc finger or coiled domain, which enables homodimerization of the NTRK kinase domain and subsequent transactivation. Returning to the figure that shows the structure of the wild-type NTRK, in the fusion protein, this whole extracellular portion, the molecule, is lost and is replaced by a fusion partner that provides homodimerization as well as overexpression. And this all appears to happen within the cytoplasm, since we don't see membrane staining in any of our cases by immunostaining, even those where the transmembrane portion of NTRK is conserved. This is data from Dr. Benjamin Durham's excellent 2019 paper, in which he presented NGS results on 55 xanthogranulomas, and he described 6 cases with NTRK fusions, as well as a range of other MAP kinase point mutations and a few other fusions. Here are the 6 NTRK fusions that they identified, and it turns out these are all solitary in his series, giving a percentage of 25% in solitary xanthogranuloma and 0% for NTRK in disseminated xanthogranuloma. Compare this to our results, where 54% of our solitary lesions had an NTRK fusion. We speculate that this lower percentage in the Durham results may be due to the targeted RNA sequencing employed in their paper, rather than the whole transcriptome method that we employed. But the overall trend is clear. NTRK fusions are much less common in multifocal and disseminated disease. We speculate that, as in melanocytic neoplasia, NTRK fusions are rather weak drivers that are common in solitary tumours, but not so effective as MAP kinase point mutations in driving a tumour towards disseminated disease. Here is the potential clinical relevance of this study. NTRK fusion xanthogranulomas appear less likely to progress to multifocal and disseminated disease. NTRK fusions are uncommon in other histocytoses, therefore pan-NTRK immunostain may have utility to confirm the diagnosis of xanthogranuloma in a histiocytic lineage tumour, and pan-TRK immunostain may have utility to screen for low-risk xanthogranuloma. This is part of our team and also part of the victorious IU faculty soccer team that defeated the residents again this year, and here are my references. Thank you very much.